Okay, Assalamualaikum and good afternoon everyone. Welcome to our first Immb International webinar series of the year. So today we have Dr. CJ Lim from the Department of Biological Sciences, National University of Singapore, to give us an insight on the structural biology from fundamental and mechanism to potential future cure. So without further ado, I would like to give the honor of introducing our respected speaker today to our IMB Director, AP Dr. Wang Siok Mui.
Thank you, Aisha. Okay, thank you, Dr. Lim, CJ, for kindly accepting our invitation to deliver this talk, your talk today. It's my great pleasure to introduce Dr. Lim. Okay, Dr. Lim is a research associate at the Department of Biological Sciences, National University of Singapore. He obtained his PhD in microbiome biotechnology from University Putra, Malaysia, where his research focused on the structural and functional characterization of virulence factors in fungal pathogens. His work integrates in silico, in vitro, and in vivo approaches, including structure-guided mutagenesis and zebrafish embryo models to elucidate molecular mechanisms underlying pathogenicity. Dr. Lim has strong expertise in structural biology, protein expression and purification, fungal genetics, and molecular biology techniques. He is also actively involved in membrane protein research, including structure-informed functional studies of G protein couple receptors, with a particular interest in ligand binding and signaling mechanisms. His research has resulted in multiple publications in reputable journals and highlights the role of structural biology in advancing fundamental understanding and therapeutic development. So without further ado, I will pass the floor to Dr. Lim. Dr. Lim, please. Thank you.
All right, thank you so much, Chair, and also Dr. Xiaomei for the kind introduction and invitations. So today I will be sharing with you on the topic of structural biology from fundamental and mechanisms to potential future cure. So I'll be sharing both my postgraduate and also postdoctoral work in UPM and NUS respectively. So allow me to introduce a little bit of myself again. I had my academic training from UPM, both Bachelor of Science and also PhD, focusing on enzyme structure, yeast biology, and in silico analysis of virus factors. So my experience includes industrial training in Singapore and research on the peptidoglycan productions, yeast biomass upscaling in bioreactors, and enzyme engineering for applications like cheese flavor enhancement. So I'm currently a research associate in NUS, studying the structural function relationship of G protein couple receptors. So this will be the outline of my today's sharing. The first part will be the in silico in vitro in vivo pipeline for validating the virulence factors in a rare Candida species. And secondly is a structure-informed in vitro functional assays of a human G protein couple receptor, GPCR. So allow me to introduce a little bit on my previous research team, where it is Enzyme and Microbial Technology Center, MTech. So there we integrated approaches like enzyme immobilization, fermentation technology, isolation, and also in silico studies to improve biotechnological processes. And it also highlights our efforts in shifting the inefficient chemical synthesis towards a greener, more efficient application such as biofuel production, industrial biomaterials, and also food biotechnology. So moving to the first session of my sharing, I'd like to introduce you about fungal infection. One of that it is candidiasis. So it is a opportunistic fungal infection caused by a fungal species called Candida species, and it can occur at mouth, skin, vagina, blood, as well as internal organs. So these infections has high occurrence in premature neonates, elders, and immunocompromised patients, and it is mainly caused by the exposure to antifungal antibiotics as well as artificial devices. So one of the Candida species caught our interest, and that is Candida gurimondi. Among all other rare isolated species, it is the most isolated one and caused the worst implications. So although Candida gurimondi was less isolated, it has been reported to cause up to 66% mortality in the clinical settings, and it mostly attributed to its increasing antifungal drug resistance. It has also been widely reported in the US, Europe, and Asia, including Malaysia. So in Asia countries also, non-albicans Candida species exhibited higher mortality. In general, fungal virulence attributes of Candida species are very complex, but they are mostly dependent on the hyphae and biofilm formation, not to forget the subsequent damage, invasion, and translocation through the endothelial cells. So the two main Candida invasion routes are induced endocytosis as well as active penetration through the hyphae that is formed. So since Candida gurimondi, or we call it Merozoama gurimondi, did not form through hyphae, the various factors such as hydrolytic enzymes and adhesive proteins are very critical, especially in the pathogenicity elicited by secreted aspartyl proteinases as exemplified in Candida albicans. So in Candida albicans, secreted aspartyl proteinases (SAP) was differentially expressed in dimorphic forms, meaning both in yeast and filamentous form. So out of the 10 SAPs, 8 are extracellular and 2 are cell wall associated. Together, they contribute to the cell wall integrity and stress response by degrading other fungal cell wall proteins, and it also initiates the adherence and form biofilm on the epithelial cells, making the yeast become less susceptible to antifungal drug treatments. So after the successful adherence, SAP is secreted to degrade the adherent junctions and mucins for subsequent invasion. In the host microbe environment, with limited nitrogen sources, the extracellular SAPs play very crucial roles to degrade the host cell proteins for nutrient acquisition and tissue damage, where the degradation of other microbial peptides and immune complement proteins is also critical for immune evasion. So here we are looking into Merozoama gurimondi, strain SO, which was isolated from spoiled orange. It could promisingly express industrial important enzymes such as lipase and amylases. However, a preliminary study showed its pathogenicity towards zebrafish embryos, but we do not know the causes. So a recent fungal whole genome has been available for this strain, where we observe 99% proteome similarity with clinical and soil isolates. So it provides a wider accessibility for us for genome editing as well as the identification of potential virulence factors. So the research design actually outlined an integrated workflow starting from the in silico identification of virulence factors in this strain SO using hidden Markov model, and followed by sequence and structural validations of the selected targets. So these candidates are then experimentally characterized through in vitro and in vivo assays, including stress tolerance assessment, biofilm formation, enzymatic activity, as well as zebrafish toxicity, alongside with the construction of gene knockouts to assess their roles in pathogenicity. So using proper hidden Markov model search, 36 hits was first resulted from 7 variance factors family. So Candida-lysine, as shaded in pink, was absent in strand SO because in Candida albicans, Candida-lysine was only expressed during hyphae formation. But as previously mentioned, strand SO does not form through hyphae. So it makes sense that this protein is not in the proteome of strand SO. So these variance factors may also present in non-pathogenic Saccharomyces cerevisiae, but their family members and expression levels might be too low to induce any variance effects. So through functionality screenings, 21 hits were proceeded for phylogenetic analysis, and our phylogenetic analysis omitted one ALS hit that stands for agglutinin-like sequences. It plays a critical role in adherence. So 20 hits from 7 variance factors families were documented in Merozyma grimondi. So we are able to detect 7 more compared to the previous individual studies. We then move on to understand the cellular localization of all these different hits. So we observed that for phospholipase B and also the protein called SSA, it is actually a heat shock protein. Their cellular localizations are contradicted with the literatures, and so the remaining 18 variance factors were subjected to further analysis. So the 3D structures of the 18 variance factors were predicted, but only 2 had suitable templates for comparative modeling, establish assay system, and conserve, as well as having high potent sequence and structural variance features. They are the cell wall associated SAP341 and also SAP1972. Through the sequence and structure analysis, both of them had similar attributes, with SAP1 and SAP2 homologs of Candida parapsilosis, which is one of the frequently isolated species of Candida. For instance, the extended CTE2, the shortened CTE2, this is C-terminal entrance loop 2, while SAP1972 exhibited an extended CTE1 and shortened NTE2. So these terminal entrance loops are very important for substrate binding and also the size of the binding cleft. Moreover, the surface areas of the druggable active site cleft in both SAP, as shown in the Armstrong here, are 2 of the largest one among all the detected members, being almost similar to the SAP5 in Candida albicans. And SAP5 in Candida albicans actually play a crucial role in systemic infections. So the larger active site cleft observed here could contribute to a broader substrate range and also multiple binding conformation of the ligands, which in turn lead to the promiscuity to the enzymes. So the deletion vectors that targeted variance factors gene were then constructed and introduced into the fungal genome. So after the successful mutants constructions, their growth were characterized and shown to be similar to the wild type. That means it doesn't affect the growth of the fungal, even though we have knocked out certain variance factor genes. And the variance reductions were then validated using the established assay. So in terms of stress tolerance, both single mutants were more susceptible to lithium chloride, where pseudohypho, as you can see in the microscopic images, was totally inhibited in the double mutant when it is treated with lithium chloride. So such observation kinds of indicates that the mutants might be more susceptible to antifungal drugs, especially lithium, to repress its virus effects. And the double mutants also show a lower tolerance towards endoplasmic reticulum stress induced by beta mercaptoethanol at 37 degrees Celsius. We then move to understanding the roles of these variance factors in biofilm mass. So what we observe is that the biofilm mass of the double mutant, as shown in the purple bar here, was 63.4% lower compared to the wild type. And it kind of indicates that these various factors contribute to adherence. And it is also shown that SAP1972 contributed more towards the biofilm mass at 34.1% reduction compared to a lower contribution of SAP341. Next, the specific proteinase activities of all mutants were significantly lowered compared to the wild type, recording up to 61.8% reduction in SAP341 single mutant. So the higher specific SAP activity in the double mutants shown in the purple bar here could be due to the compensatory upregulation of other SAPs, which we are not so sure because we didn't really do regulation studies like qPCR to check whether it is really so. But this observation has been reported in other investigations in Candida albicans. So lastly, our in vivo study actually showed the signs of various endpoints such as short tail yolk sac edema and pericardiac edema, where these signs were less severe in the embryos or larvae treated with our mutants. So at 24 hours post-exposure to the mutants, the fungus, SAP1972 mutant, and also the double mutant showed reduced biofilm formed on the chorionic wall of the embryos. So it justifies the biofilm formation studies in our previous essay. And biofilm was initiated by adherence. So it is kind of important because without the adherence, it is quite difficult for Candida to cause the infection since it's the first stage. So we also look at the fungal burden, and the data actually suggested a lower invasion rate in all the mutants. It hypothesized a higher fungal clearance by the immune system, the innate immune system, showcasing the crucial roles of biofilm formation and SAP activity in adhering and invading the embryos, as well as degrading the immune complement proteins for immune evasion, respectively. We then check on the mortality rate. So in agreement with the previous observation, the mutants showed up to 62% reduction in the mortality rate compared to the wild type at 96 hours post-exposure. So the mutants showed reduced virulence compared to the wild type. So in conclusion for the first part, the variance roles of SAP341 and SAP1972 were successfully elucidated via the constructions of structure-guided mutants through the in silico, in vitro, and in vivo pipeline. And for this part, I would like to give acknowledgement to my supervisor during my PhD studies, Associate Professor Dr. Siti Nobaya Oslan, as well as all other supervisory committee members and co-researchers, as well as the lab members of in MTech and Institute of Biosciences. So this ends the first part of my sharing. So the second part of my sharing, because some of the slides might have data which are pending publications. So here's some disclaimer that it will be great to not recording this or to take photos of it because of the intellectual property issue. All right. So a brief introduction on what we are doing here is on structural biology.